Hi guys, this is Mujtaba Ahmed and I am back with 8th lecture in this series on O-levels biology. Before today's lecture, I'd like to share a short story about my childhood. And back in my childhood, when it used to rain, especially in the seasons of monsoon, after the rainfall, we used to see these slugs and snails that you see on your screens right now popping out and coming out of their homes into our car garage and porch as well. But in order to get rid of them, as a home remedy, we were told to spread or sprinkle table salt that's readily available in every home, I guess. So what used to happen and what we used to observe was that as soon as the salt was sprinkled onto their bodies, the snails and the slugs used to shrivel, contract, and within no time they used to die on the same place, on the same spot. So I had no idea that there was really some good reason or science going behind that mechanism as well. But it turns out that the phenomena of osmosis that we also discussed a little bit about in our last lecture is responsible for their death, for causing them to dehydrate actually and shrivel and then die ultimately as a result of spreading salt on their bodies. So what happens is that spreading salt really sucks water out of their bodies by osmosis, because osmosis, remember, is the net movement of water, and to balance the concentration of water outside compared to inside. The water moves outside onto their bodies, and they as a result dehydrate and shrivel and die as a result. So it was really interesting for me to know and to relate this to my past childhood experience when I was preparing for this lecture, and now we will move forward with the learning outcomes of this lecture, and these are the things that we are going to understand in this lecture. So the first one is that we are going to understand that how important it is that the water pressure be maintained inside the cells to maintain the complete plant body. Then we are going to discuss the effects of osmosis on plant and animal cells and animal tissues, and explain the importance of water potential gradient and osmosis in the uptake and loss of water. So remember where it mentions explain, we are looking for a reason, for a convincing reason to the examiner, because he can ask any reason and it can be looking for that reason, particularly reason that is responsible for a phenomena in context. And then we are going to investigate and explain the effects on plant tissues of immersing them in solutions of different concentrations, and remember we would be using the terms turgid, turgor pressure, plasmolysis, and flaccid. Remember, these are the terms that have been particularly mentioned by the examiners or by the Cambridge in their syllabus copy. So you must be able to use or refer to these terms, and must be clear about these terms as well. Then we are going to investigate osmosis using materials such as dialysis tubing. It's a little and straightforward experiment that I'll summarize as well. And then explaining the importance of active transport in ion uptake by root hair cells, and the definition obviously of active transport would also be taken up in this lecture, towards the end rather of this lecture. So really it is going to be an interesting one, and an easier one as well, I should say, to grasp on. So the first slide of today's lecture talks about the importance of maintenance of pressure of water inside a plant cell. So remember that we discussed that plant cells are made up of cellulose cell wall. So cellulose is a biological molecule, and it has OH bonds, OH molecules, or hydroxide molecules, OH molecules, and because of this they are able to bond a sort of bond which we call a hydrogen bond with water. So it's a particular bond, sort of bond, which exists between the electronegative of one molecule and hydrogen. So hydrogen is contained within this water molecule. So hydrogen is partially positively charged and oxygen is negatively charged. So a strong bond, so to say, can be formed between such molecules. Just remember this. And then this is because of the oxygen atoms, as I told you, in the OH group and cellulose can have hydrogen bonding interactions, which is a really particularly strong force, which allows the bonding between hydrogen and OH of cellulose. So water basically exerts pressure towards outer side towards the stem, and this pressure is maintained throughout the stem and throughout the plant body. So we have been using we'll be using this term turgor pressure. So it is important to understand that it is a pressure that is exerted by fluid. In this case we are discussing about water in particular. So this is the pressure exerted by water onto the plant body or against the cell wall particularly, and it's also referred to a hydrostatic pressure. Hydrostatic because hydro means water, hydro is pertaining to water, so it's a pressure due to the presence of water. You have to understand, so I place this diagram. This diagram A shows a wilted plant which shows drooping leaves, okay. Because it has lost water, it has not completely lost water, but it has lost its turgor pressure, so to say, to maintain its turgidity, or so you to say fresh effect of the leaves or the plant, in comparison to this one on what you see as B, plant B. It has complete turgor pressure. It's not a flaccid plant. Flaccid means that it's not led to wilting, so to say, and it's a fresh plant because it has a maintained and ideal water pressure. So it's very important to understand that that water pressure must be maintained throughout the plant to give it a fresh look, to give it a really good look as well, and for it to maintain its body as well. So that's very important. And we're going to talk again about the terms flaccid as well. So this is a term flaccid. This is a flaccid plant because it has lost its turgor pressure. So we are going to talk about these terms, flaccid, turgor pressure, plasmolysis as well in the coming slides as well. So what is water potential and osmotic pressure? Because it is important that there exists water potential for osmosis to take place. Remember that osmosis is the net movement of water from an area of higher concentration to where it is lower in concentration. So when the pressure from the fluid involves water as a result of influx across a semi-permeable membrane, it is referred to as osmotic pressure. So it's the influx across a semi-permeable membrane. So it is the movement inwards across a permeable membrane to a medium where the water concentration is lower. Because of the differences in solute concentrations between solution, water diffuses to where there are more solutes. So this is similar to what I explained just in a moment. This tendency of water to move from one area to another is called water potential. So it's the definition of water potential. It's the tendency or the ability of water to move from a higher concentration or from one area to another area to maintain equilibrium. It's called water potential. When water moves into the cell becomes turgid. This condition wherein the turgidity is maintained, so turgid or swollen, is called turgidity. So turgidity is the term we use to explain or we use to refer to a plant cell where it has enough water or its plant cells are swollen enough. So it's turgidity. Now we talk about the types of solutions, because we are going to compare the animal and plant tissues in various scenarios, and in these three scenarios particularly. So isotonic is the first one, and it's the really simpler one. It's the solution. Iso means same. Remember this word. Iso meaning isomers, you are going to learn in chemistry class as well. Isotonic means same tonicity, so it has same solution, and this is what the definition pertains to. same concentration of solutes both inside and outside the cell. So understand that the concentration of solutes in a solution in two places, so to say, are exactly same. So there is equilibrium already maintained in that case. For example, a cell with the same concentration of salt inside it as the surrounding water fluid would be said to be an isotonic solution. Under these conditions, there is no movement of solvent. In this case, the amount of water entering and exiting the cell's membrane is equal. So remember that at equilibrium, I told in the last lecture as well that there is a movement, but no net movement. Net movement means that there is no loss of concentration or no alteration in concentration, but the movement of water from one side or from one region to the other is remains same, and therefore equilibrium is maintained because there is no alteration in the concentration to the either side of the two or many places. In a hypertonic solution, however, there is a higher concentration of solutes inside the cell. This means that inside of the cell is more concentrated with solute than the outside cell. When this occurs, more solvent will enter the cell than leave it to balance out the concentration of solute. So it's natural to understand, so it's very easy to understand that water or the solvent will move from where it is higher in concentration to where it is lower in concentration, because higher concentration of solutes equal to lower concentration of solvent. It's similar to saying that. So lower concentration of solvent, so the solvent will move from a higher concentration of solvent to a lower concentration of solvent. A hypertonic solution is the opposite of a hypotonic solution. There is more solute outside the cell than inside it. So the inside of the cell is less concentrated, or it's dilute compared to the outside. It is this type of solution, in this type of solution more solvent will exit the cell than enter in order to lower the concentration of solute outside the cell. So remember in the starting we talked about how sprinkling of salt would lead to the movement of water outside of their bodies. This is the same scenario that's happening. The water would move out to balance the concentration outside. Concentration outside. So this is pretty much straightforward, and these are the definitions, complete definitions that I have mentioned and included on this slide. So you need to just practice and try to grasp the understanding and try to put these definitions inside and grasp them in your mind as well, and it will be really straightforward and easier, I guess. So now we'll be comparing the animal cell and plant cell, which is on the downside of this diagram, to see how they behave in different scenarios, in different sorts of solution. Remember the hypertonic solution, isotonic solution and hypertonic solution. We just discussed these sorts of solutions right now. So in case of a hypertonic solution where the concentration of solutes outside is more and the water will tend to move outside as a result, in case of, so these are red blood cells, guys. So for reference we have we're talking about RBCs, which are the red blood cells in animal cell. So these cells are, the animal cells are seen to shrivel or shrink because they have lost their water to the surroundings or to the outside. Because the water potential was higher inside compared to outside, and water moved from the inside of the cell to the outside of the cell. And in case of let's talk about what happens in the case of the plant cell. So in case of plant cell, the plant cell would become plasmolyzed. It's important that you note this term and use this particular term as well in your exam. So so the water has, it's showing that water is moving outside towards the surroundings, and what you should note is that cell membrane, this is a cell membrane of a plant cell which is seen to be shrinked as well, because remember that cell wall remains unchanged. Okay, Cell wall is a structure which is particularly hard. It's a cellulose cell wall, and it's very solidified structure, and it won't be affected by the really by the varying concentrations of solvents and solutes. So if you were to ask to draw a diagram, you must be able to differentiate this, because so you would draw a little star like ignore my trying, guys, shape. So this is the cell wall, which will remain unchanged. This is the cell membrane. Okay, you have to show this to the examiner. The cell membrane would lose its shape because it's not a rigid structure, but cell wall would stay the same in every case. Note this, in this case, in the case of flaccidity and then turgidity, sorry, the cell wall shall remain almost same, but in this case it's slightly altered, and that's because of the extensive pressure of water onto the cell. But all in all, what you should observe is, at least in these two cases, the exact same shape is observed. Rather, cell membrane is the structure that you should be very keen to notice and observe, and show this on your diagram as well. So in case of isotonic solution, where the concentrations of solutes and solvents is not changed or is relatively constant, it is the ideal solution for animal cells, so it would maintain its shape. However, a plant cell would become flaccid because it will be losing water and gaining water as well, and it's not really an ideal situation. The ideal situation for a plant cell is in case of turgidity, because it keeps the plant cell walls pumped, so to say, or maintains throughout the plant body the pressure, as I told you in the beginning. So plant cells want to remain some sort of swollen, or they need water to exert pressure on their cell walls to remain erect as well. So this is the ideal situation. I've read this, that ideal situation for the plant cell is turgidity, and for an animal cell it's in case of isotonic solution. And remember that cell wall is an important feature of a plant cell wall, which avoids it to get burst, because we see that in hypotonic solution an animal cell has got burst. Okay, so you understand this difference is because of the presence of cell wall in the case of a plant cell. This is a little experiment, little experiment, because it's very straightforward, and you have to understand the crux of this experiment, because it would be mostly asked in P1 MCQs part, and it's very straightforward. This is just what we discussed, and this is really the application of what we have discussed. So this this is a beaker of water, and this is a whisking tubing into which we have poured a sucrose solution, so to say 10%, I guess, yes, 10% sucrose solution, and then capillary tube which measures the level of liquid, which would rise or fall depending upon the scenario. So in the first case, this table describes the different scenarios. So in the first case, water so what happens is, sorry guys, so what happens is that we have to see what is the pressure of what is the concentration of water inside and outside of the cell. So liquid outside, if liquid outside is water and liquid inside is 10% sucrose solution, in the first case we are discussing this case, what would happen is that water would sorry, because water is more concentrated outside, the water would move inside. Okay, and as a result, what we would observe is a rise in the capillary tube because of the movement of water inside. If the liquid outside is 10% sucrose, this means that concentration outside compared to inside of a whisking tubing or this dialysis tubing, we can call it as well, or we can refer to, because it's similar to that similar in functioning to that. 10% sucrose solution is outside the whisking tubing, and liquid inside is water. Then the water would move from inside to outside, because water is water is moving from an area of higher to an area of where it is lower in concentration, and the solute moves the movement of solute would be outside to inside. So it's exact opposite. See, these are the exact opposites, and liquid level would fall. If water is outside and water is inside, then there would be no net movement and no visible change. And similarly, if both sides are filled with 10% sucrose solution, then an equilibrium is maintained and no change is visible in these two cases. So it's important that you understand this table, because this can get a little tricky, I guess, but you have to understand that the water will move from an area of higher to an area of lower concentration. This is just an application of the concept of osmosis. Now, just don't get scared by the amount of words or the sentences that you see on the slide. I just put them knowingly, because this is where you have to explain the significance of ATP, and which is active transport. ATP is another thing, active transport, and its significance especially in reference to the uptake of ions in case of a root hair cell, which is a plant cell. So active transport is basically a type of cellular transport mechanism in which substances are moving from an area of lower to an area of higher concentration. So it's the exact opposite of diffusion, but it is important that you note it requires energy. So for an ion or for a biological molecule or any molecule to move against its concentration gradient, it requires energy, and energy comes from in the form of from the mitochondria in the form of ATP. Remember, we talked that mitochondria is the powerhouse of the cell, which provides it with this molecule ATP, adenosine triphosphate. It's a biological molecule which is synthesized at the sites of mitochondria, and it's required for the energy for the various cellular functions and various cellular mechanisms. So ATP provides a cell with this essential energy to move or uptake ions or molecules which are lower inside of a cell. So this was the definition which you see on your slide. This is the definition: from an area of lower to an area of higher concentration. Hence, this will require expenditure of energy in the form of ATP, and it can also require the assistance of membrane proteins such as carrier proteins as well. So there are broadly two types of active transports, but we don't need to get into that detail. Primary active transport and secondary, which requires the coupling of molecules. Then in this concept it also mentions uniporter or symport transporters, and that's not really the part of your syllabus, but I'm just telling you there are broadly two types. So plants can only absorb soluble minerals, so which are the minerals which are dissolved in water. So they absorb minerals dissolved in solution from the soil through their root hair cell. So this is a particular type of cell which is responsible for the absorption, and its structure is perfectly suited to its function. It's like a long sort of elongated sort of cell. It seems like this, and it has root hairs to increase its surface area for absorption, for maximum absorption from the soil. Okay, this is a root hair cell. Maximum absorption, for the maximum absorption of minerals or ions from the soil. So the concentration, it is important that you understand that the concentration of minerals in the soil is very low, but because osmosis is the net movement of water from an area of higher concentration, or it's rather the movement of water only, um, it cannot be absorbed by the virtue of osmosis. And because diffusion involves the movement of substances from high to low concentration, it can also not be used. Instead, uh, the nature has this alternative process or cellular process, which is what we call active transport, which allows the movement of ions or minerals from an area of lower to an area of higher concentration in the root hair cells. So root hair cells have carrier proteins in the cell membranes. As I told you, that multiple cells also carry or uptake ions by the presence of these special transport or carrier proteins. These pick up mineral ions and move them across the membrane into the cell against the concentration gradient. Because active transport moves against concentration gradient into the root hair cells, energy is needed. The energy supplied in the form of ATP. So it's adenosine triphosphate. It's the molecule which is synthesized at the sites of mitochondria, as I told you, and it's an important molecule for providing cell the essential form of energy for the various cellular processes. So guys, this was it. This was really about the complete chapter, or the complete complete topic of the movement of into and out of the cells, and it covered it began with the diffusion, the factors that affected the rate of diffusion. Then we talked in the previous lecture about the osmosis as well, and in this lecture we continued with the osmosis. We also discussed the types of solution, the behavior of animal and plant tissues when kept in different solutions, the isotonic, hypertonic and hypotonic solutions. And then we also investigated the little experiment that I discussed with the whisking tubing, that really tests the applications of osmosis. And towards the end of this lecture, we have discussed the importance of active transport, especially with reference to the root hair cells and how it allows it to uptake ions from the soil. So this was all about the topic, and we have ended this topic. And please let me know if you have any queries or suggestions. I'd like to address them, and I'd like to improve myself as well. Till next time, take care, happy learning. Thank you very much.